I'm really delighted to introduce our keynote speaker for today, Dr. Devon Crawford. She is the Program Director in the Office of Research Quality at the National Institute of Neurological Disorders and Stroke, NINDS, at the National Institutes of Health. Devon works primarily to improve biomedical research culture, to better value and implement experimental rigor and transparent reporting. So you can see, very aligned with our mission here at BITSS. She received her PhD in neurosciences from Washington University in St. Louis and completed postdoctoral research at the University of Texas Southwestern Medical Center. I personally had the pleasure of getting to know Devon a couple of years ago at an NIH meeting in Maryland, where we discussed our common interest in research transparency and open science, and I got to meet others within NIH who for years have actually been championing these ideas of promoting open science, credibility in research. So it's a great honor to welcome her to our community and hear her perspective, and let's give it up for Dr. Crawford.
Thank you. All right, I was told that the microphone would just automatically turn on. All right, thank you. Okay, thank you so much for that introduction. It was very lovely, very nice. And thank you all for having me. Move to my slides, yes. Okay, so I was tasked with discussing rigor, transparency, and replicability, sort of writ large in the context of NIH as a way to kick off this meeting. And I will say that I think this is a very important topic, but I probably have my biases given the work that I do. Which reminds me that I do need to start off by saying that any opinions I express are my own and not those of NIH, HHS, or the US government. So I'm speaking in my own capacity here, all right? So I'm coming at this as a member of, as he said, the NINDS Office of Research Quality. And our general mission is to promote experimental and analytical rigor, measures to reduce bias, transparent reporting, and high-quality scientific research. And as of today, at least, our office is 3 people strong, so it's just myself and 2 other people. So we're this very small team trying to steer a big ship. But we care very much about this mission and want to do what we can to improve research quality. But with that context, we are gathered here today for BITSS 2026. And this is just advertising I took off the website and from the emails. And it's very clear that, you know, the theme of today is really about AI, open science, LLMs, data quality, privacy, transparency, and all of those good things, which is not necessarily my area of expertise. So it's a pretty tall order, and I have no doubt that the other speakers today will be able to very much rise to the occasion, but it had me asking myself, what could I potentially contribute to this conversation as a more biomedical researcher? And of course, in the biomedical sciences, we are very much excited about the leaps and bounds being made in computational modeling and AI. You know, as I said, it's not really my area of expertise, but I look forward to learning from everyone today. And in this, you know, brave new world, as I labeled it, there are lots of new opportunities to be able to do things we couldn't do with human brain power alone. But of course, that comes along with its own caveats and responsibilities. You know, we already discussed some of those in the opening, you know, open science, data sharing, code sharing, data privacy, security, all of those things, right? And one sort of guiding standard in this space has been FAIR, the fair data, fair code, findable, accessible, interoperable, and reusable. And NIH also promotes the FAIR standard. And this is just sort of an excerpt from the data management and sharing plan that you have to submit when you apply for an NIH award. And it's currently undergoing some updates. They're sort of taking out some open text things and replacing them with yes/no answers. But you still have to talk about what data you're collecting, what file types, what repository you're going to be putting those in. And so when it comes to FAIR data, though, the devil can really be in the details, right? You know, we talked about these issues of security and things. But when I think about this, it's like we really are just focusing on one part of the research process, and that's sort of the transparency of data handling and analysis. So what you're doing to the data after they've already been collected, which is super important. But I like to think about, well, what happened before that? How are the data collected? How are the experiments designed? Where did these data come from? What caveats might be in there that you need to know about? And as this paper explains, although fair data are necessary for reusability, this does not guarantee scientific rigor, trustworthiness, or research quality. Something got messed up with the formatting there. And as they say, garbage in, garbage out, right? And so metadata, other information, really does need to include the full experimental protocol and other information like that for it to be truly useful. Because if we ignore rigor, then all of the effort put into collecting those data and analyzing them could themselves have been wasted. And this is where I want to shout out session 2 today, improving research quality, because I think even though you didn't get top billing, this is a really important part of the research process. But bringing it back to biomedical research, we have what's often referred to as this valley of death between academia and industry, where it's really hard to get from initial biological discovery all the way to approved clinical treatment. This can take many years, billions of dollars, and still only be successful like 0.1% of the time. And there are, of course, many reasons for that, some of which are like regulatory, time, funding, but highlighting at the top is what this paper labeled as reproducibility, and I put an extra label of rigor and transparency, because I'll get into later how rigor and transparency are really a prerequisite for the kind of reproducibility they're talking about, and reproducibility by itself doesn't necessarily stand on its own. But rigor and transparency are something that we can control in the laboratory. So this is the design and execution of our experiments. And this is how we often think about our experiments, is sort of in this cycle where we generate our hypothesis, we design our study, we conduct it, we analyze it, we interpret it, we publish it, right? And then we use that to design the next study. But at each of these stages, less rigorous practices can creep in to help create spurious results. For example, if we fail to control for bias, if we have low statistical power, poor quality control, p-hacking, hypothesizing after results are known, or publication bias, and a lot of these things I think will actually get discussed today, then these can work together to help weaken the strength of evidence in the scientific literature. And all of this can happen without any sort of like fraud or misconduct. And so rigorous experimental design, in my mind, really comes down to trying to reduce experimental bias. And this was described by David Ransohoff in this commentary as unintentional and unconscious. So we're not talking about intentional bias, misconduct, or anything like that here. It's defined broadly as the systematic erroneous association of some characteristic with a group in a way that distorts a comparison with another group. But we all have unconscious biases that we bring to our interpretation of the world. Francis Bacon recognized this, writing in 1620 that the human understanding, when it has adopted an opinion, draws all things else to support and agree with it. And though there be a greater number and weight of instances to be found on the other side, yet these it either neglects and despises, or else by some distinction sets aside and rejects, in order that by this great and pernicious predetermination, the authority of its former conclusions may remain inviolate. So in other words, we tend to confirm what we already believe to be true, right? And having this unconscious bias doesn't make us a bad person or a bad scientist. It's just a part of being human. What matters is what we do to navigate those biases, to try to do what we can to get closer to the truth of a phenomenon. And I also like this quote from Epictetus in the 2nd century, saying that appearances of the mind are of 4 kinds. Things either are what they appear to be, they neither are nor appear to be, or they are and do not appear to be, or they are not and yet appear to be. Rightly to aim in all these cases is the wise man's task. So how do we aim rightly? Well, at NINDS in 2012, we actually held a workshop and a bunch of scientists and publishers were in attendance. And one of the outcomes of that workshop was a paper where we outlined 4 practices that really should be reported for all preclinical research. Those are blinding, randomization, sample size estimation, and data handling. And data handling here means more like inclusion-exclusion criteria. It's not just data analysis. And these are often referred to as the Landis 4, after the first author of the paper, Story Landis, who was NINDS director at the time. So why do we care about these 4 practices specifically? Well, they help reduce those biases that I talked about. This is an example from related to blinding, where they had a systematic review of human studies, and they looked at the effect size in blinded and unblinded assessors in the same studies. And they found what is essentially an increased effect size when they were unblinded. So they wrote that in 63% of the trials, the effect size point estimate was more optimistic, means larger, as determined by the non-blinded assessor. The standardized mean differences were exaggerated by a pooled standard deviation of 0.23. And so unconscious biases here could have potentially skewed the results when they were unblinded. And as another example, just thinking about sample sizes, we all know that often sample sizes are too low, people aren't doing power analyses, that's definitely a big issue, but also sometimes people aren't thinking about the full context and consequences of what can happen when you have low sample size. So in biomedical research, we try to have an 80% chance of detecting a difference between groups when one exists, but actual power tends to be much lower than that. And so in this example, they took 660 meta-analyses. They used the effect size in each meta-analysis to sort of back estimate what the power was in the original studies that went into it. And you'll see that about half of the studies had less than 20% power to actually detect a difference between groups. And so power was much lower than people actually thought it was. And this can cause lots of issues, as you can imagine. And people often have, I would say, an optimistic estimate of their effect size when they're going into power analyses. And so this could be one issue. And this low power is a shame, because imagine all of the effects that they're not able to detect because they have low power. But there are also other issues going on when you have low power. And that's shown here when they graphed effect size versus sample size in the exact same studies. And you can see that there is a negative correlation there. So when they had low sample size, they actually saw inflated effect sizes. And this can be due to things like a publication bias, where if you have a low sample size, you're really only going to get published if you have an exciting result, but also due to the winner's curse, where if you are underpowered, you tend to only see statistical significance if you have, in fact, overestimated the sample size. And so being underpowered can actually lead to this bias in the apparent results. And this is why it's not always advisable to do a pilot study to get your estimated effect size before using that in a power calculation for a full study. It's because you can have an overestimated effect size that then gets used in the power calculation, and then you've created yourself an underpowered study. And this is definitely a big thing in biomedical research. All right, and I could give you multiple examples of this, but this slide is really just trying to make the point that these things can combine. So this is a systematic review of, I believe, like 109 experiments from about 24 preclinical stroke papers, and they asked how many of these quality characteristics were present in each of the papers. And those were listed down on the left, and they include things like the Landis 4, like blinding randomization, but some other things as well. And none of the papers came close to having all 10 elements, but you can see this negative correlation. And so by not having these practices that are there to prevent unconscious biases, then we can actually get these combined effects to get bias in the apparent outcomes. So coming back to this valley of death, I sort of promised you a link between lack of rigorous research practices and a failure to translate from the bench to the bedside. And so I'd like to talk about this case study, which involved mouse model of ALS, so the motor neuron disease. And there was some promising results in the preclinical studies. And so there were clinical trials that actually went on to fail. And those drugs are listed down the left side here. And in the blue bars that go off to the right, that's the increased survival that was seen with each of the drugs in the original studies. And as I mentioned, these failed in clinical trial. So the ALS Therapy Development Institute came along and did new studies, but here they implemented blinding of investigators, randomization to treatment group, made sure to have sufficient sample sizes, so more like 24 per group rather than the typical like 8 mice per group that are often in these studies. They implemented sex and litter matching. They made sure to predefine when they were going to take data out of the data set, and they transparently reported these items. So a lot of this stuff was missing from the original studies. And what they found is in the dark bars. In every single case, there were small and sometimes negative effects of each drug on survival in this mouse model, which actually much better matches the clinical trial. And so by not having these rigorous research practices, we got invalid results that led to completely unnecessary clinical trials that then went on to fail. And so that's why this is so important. And yet, despite how important these practices are, there seems to be some hesitancy in actually reporting whether or not they are used in biomedical research experiments. So this paper looked over 2 million papers from PubMed. They used a text mining algorithm to check whether or not some of these practices were even mentioned. So whether they did it or not, did they report it at all? And despite some gains over the last 20 years, you can see that blinding, sample size estimation top out at about 10% of papers reporting, and randomization and replication about 30%. So it's still pretty low. Where was I? That's right. So because it's so low, we don't know if this is really due to people being unaware that this is important or if it's something else. But if it is awareness, that's a pretty frustrating plateau because there have been reporting guidelines available for a very long time in multiple different research areas and types of experiments. You have, for example, the ARRIVE guidelines for animal studies, CONSORT guidelines for clinical trials, as well as STROBE guidelines for I believe it's epidemiological type observational studies. And the ARRIVE guidelines even underwent a recent update to try to get down to a top 10 and essential 10 that are the basic minimum that without which you cannot assess the reliability of the findings. And this update was made entirely to make it easier for people to report these things. And there are even journals out there that require adherence to these reporting guidelines, and people give assurances that, yes, I followed the reporting guidelines when they publish in journals, and yet this still seems not to have solved the issue. But I don't want to imply that I don't value research that cannot inherently report these practices. There are, of course, things like exploratory research, where you're not asking a specific question. You might just be observing what's happening. And these are still super important questions because they can drive you to new questions. And in this case, it's really good if the experimental design elements can be incorporated where possible, but it might not be possible. And in that case, we just need to have more caution when we're interpreting these experiments. What I'm really talking about today is the more confirmatory type experiments. And these are asking a specific question. They might, for example, be looking for a causal relationship. And in this case, all applicable elements really should be incorporated when physically possible, essentially. And this is because we want to make important decisions like, should we go to clinical trial? But what's most important is to be transparent about reporting these things, whether or not something was done. And this sort of transparency can actually be really useful when we're trying to combine data. For example, with systematic reviews of health studies. And in these systematic reviews, there's always a risk of bias assessment, asking, did they blind? Did they randomize? Did they have your complete outcome data? And for each practice, they assign low risk of bias, high risk of bias, or some concerns if it's unclear. And the overall risk of bias from a paper is determined by how much of these high-risk items are there. And you can see that this is done for every study in a systematic review. That is just an example down there on the bottom right. And so this can then be used to weigh the data appropriately in the meta-analysis. because if something has a high risk of bias, it doesn't really make sense to weigh it equally as something that has much stronger evidence. And so going back to this flow diagram, I do very much care about this stuff. I do care about transparency. But it's not just code, data, analyses. It's also what went into collecting those data before you ever see the data, to know whether or not there's some sort of risk of some underlying flaw that could then lead to all of your analyses also being flawed. And one thing that we have been piloting at NINDS to sort of try to improve transparent reporting, especially at conferences, is the use of what we're calling Rigor icons. And this did not show up at all. Is this not the PowerPoint version of the slides? Well, the PowerPoint doesn't have all the formatting issues that the PDF has. But there's supposed to be some lovely icons in this table here for you to look at that maybe will be available after the conference, that file. But we made icons for blinding, randomization, sample size estimation, data exclusions, as well as whether or not an experiment was exploratory, because maybe that would explain why something might not have the other practices. But okay, it kind of shows up here. So, and the way that these would be used is like so. You would take some sort of minimally informative data panel, like what you see on the left, and then you could turn it into something a bit more transparent on the right by adding things like the sample size, the spread of the data points. And at the bottom, you can see these icons for randomization, blinding, and saying it was a confirmatory study. And so this gives you much more information when you are giving presentations, for example. And early pilots with this have suggested that when people are using them, it at least gets them to think a little bit more deeply about their experimental design. Often it's too late for them to change anything about their experiment, but hopefully that gets them to think about doing it a little bit better the next time. And of course, you know, we in our particular field feel like the Landis 4 are the most important, but you know, in other fields, for example, each of your fields, there might be other practices that you would consider super important to report. But one reason I bring that up is another way that we could potentially have better reporting of these practices in a useful way in data science is through the use of common data elements or CDEs, or potentially metadata. So NIH actually has its own CDE repository. Just to make sure we're on the same page, they define CDE as data elements or variables that are defined and used the same way across multiple studies to standardize the way data are collected. And metadata, of course, information about other data, but when you're in the space of experimental design elements, sometimes the boundary between CDE and metadata gets a little fuzzy, so I'm just sort of using both terms here. But the CDEs in the NIH repository are really focused on clinical studies and not necessarily preclinical studies, which is where we see most of the issues. And it's only recently that they've been sort of expanding into the preclinical arena. And one example of that is what's called NT Proceeds, which is being led by NINDS, and that stands for Neurotrauma Preclinical CDE Data Standards Program. We do love our acronyms. And just full transparency, I'm on the steering committee for NT Proceeds, but what they're doing is they're bringing together researchers in 3 different areas, and that's traumatic brain injury, post-traumatic epilepsy, and spinal cord injury. And so these are preclinical research areas to put together some CDEs, but you'll see circled in red is one working group that is specifically devoted to developing CDEs or study metadata related to rigorous experimental design elements like the Landis 4. And so if these could become more regularly collected and shared alongside these data in repositories, then it could become much easier when analyzing and weighing data sets in the future to be able to do it according to that underlying risk of bias. That is not how that's supposed to look, but that's okay. But compounding everything is publication bias, and I know we'll be talking a bit more about that later today. And this is sort of the ultimate lack of transparency, because you're putting all the effort into doing an experiment and then deciding whether or not to disseminate it based on the results. So, you have a statistically significant effect, it's super exciting, you're gonna put all the work into trying to get that published in a high-profile journal to get that career advancement. But let's say it's not statistically significant or it's not exciting, it might just get filed away. And so because of that, a lot of knowledge can get lost despite all the effort that went into creating that knowledge. And I know you're all probably super familiar with this example, might even come up later today in the social sciences, where they looked at over 200 papers from an NSF-funded program. And those that had quote unquote strong results or all or most of the hypotheses were supported by the statistical tests. Over 60% were published and only 4% were never written up. But when it had mostly null results, 65%-ish were never even written up and only like 21% were published. And these all had very similar methods. They were all considered rigorous enough to fund. And so it's not like, oh, the null studies were just not rigorously performed and therefore they don't deserve to be disseminated. So it appears that the outcome probably was some sort of driver in determining whether or not it got disseminated. We have very similar issues in the biomedical sciences. So in this example, there were 74 clinical trials for 12 antidepressant medications. And each column here references a particular medication. And if you look just at the published studies, that's at the top. Out of the 74 clinical trials, only 51 ever got fully published. And of those, only 3 had null results. But if you look what was submitted to the FDA about those clinical trials, that's at the bottom. You can see that only 38 were actually positive, not 48, which we'll get to in a second. And there were 36 null studies, only 3 of which were reported as null studies. But 37 out of the 38 positive ones got published. So almost all of them got published when they were positive. Interestingly, 11 of them were null as assessed by the FDA, got published as if they had positive results. So that's not very transparent, I would say. And again, this isn't just due to a lack of rigor, and therefore it shouldn't be disseminated, because these are randomized controlled trials that were submitted to the FDA as evidence. So there's no reason these shouldn't have been published. But it does skew what the effect of these drugs looks like, because if you look at the published studies, it looks like 94% of them are positive. But really only 51% are. And it also inflates the effect size. The apparent effect size goes from 0.31 to 0.41, which could affect future power calculations, as we discussed earlier. And so these drugs look much more effective than they actually are. And in this way, publication bias can really harm the integrity of our collective scientific knowledge. And Ted actually mentioned this already, but we have an interest in this at NINDS and held a workshop in 2024 specifically on this topic, novel approaches to preventing publication bias. So it was great having some of this community there. And the general feedback that we got from the participants was, one, that career incentives really should focus on quality and process of the work and not just what the outcome happens to be or what journal it gets published in. Statistical significance should not determine whether or not something gets disseminated, and all members of the scientific community really need to contribute to trying to change the incentives to promote dissemination. And I will say that the current NIH director, Dr. Bhattacharya, has at least stated in public meetings that one of his shared goals is to try to make null findings more discoverable, findable, and accessible. And so we'll see what happens with that. But as sort of an aside, even the statistical community criticizes this practice of not sharing research based on the outcomes. And in 2016, the American Statistical Association said there's so much confusion around p-values and statistical significance, we're going to come out with a statement. And they got together dozens of statisticians who were able to agree on just 6 statements, which I'm impressed they could get them to all agree on 6 things. But the first thing is that the only thing p-values really can do is indicate how incompatible data are with a specified statistical model. They cannot measure the probability that a studied hypothesis is true or that they were produced by random chance alone. Again, only looking at compatibility of data with the model. They should not be used for important decisions, like whether to disseminate work, based only on whether it passes a specific threshold. Proper inference requires full reporting and transparency, so we can't just be cherry picking what to report. And a p-value or statistical significance does not measure the size of an effect or the importance of a result. So if you have a really low p-value, that doesn't necessarily mean it's a more important result. And this is because p-values are sensitive to things like our sample size and the precision of our measurements that might not actually have anything to do with the underlying effect that we're looking at. And by itself, a p-value does not provide a good measure of evidence regarding a model or hypothesis. Because one, there are other models or hypotheses that you could have been testing. But two, it doesn't say anything about magnitude of the effect, confidence intervals, or anything like that. And so we probably shouldn't be putting so much power in the hands of a p-value here. And that advice should probably not be all that new or surprising to us, because influential statisticians like Ronald Fisher, who's complicated historical figure, we're putting that aside for right now, have been saying this all along. In 1935, he wrote that if we accept this convenient convention of this statistical significance threshold, we thereby admit that no isolated experiment, however significant itself, can suffice for the experimental demonstration of any natural phenomenon. For that, one chance in a million will undoubtedly occur with no less and no more than its appropriate frequency, however surprised we may be that it should occur to us. So in other words, a single p-value from a single experiment actually tells us very little. And he goes on to say that if the design of an experiment is faulty, any method of interpretation which makes it out to be decisive must be faulty too. So if it's not designed well, then a p-value or any analysis based off of it is fairly meaningless. And this is because rigorous experimental design is a foundational prerequisite before ever even trying to attempt any sort of statistical inference. And that reinforces sort of the message of what I've been trying to say today. I didn't want to leave before talking about this last step. I'm not totally ignoring it, and that's repeating our experiments. This is your reproducibility, your replicability, your robustness, your generalizability. And this is, of course, super important because you need to know whether or not the observation that you're making is, it actually exists and can be found over and over again reliably. And this topic has sort of had a resurgence in interest, I'll say, recently. It's also one of Dr. Bhattacharya's stated priorities, although this is something NIH has been doing for many years, is working on this problem. In the top left is a website we've had for a long time showing that NIH has long promoted reproducibility. And here we're using the broad definition of the term. So it's more like generalizability and replicability. Can only really be attained through rigor and transparency. Because as we discussed today, you can replicate biased results really well with less than rigorous methods, right? But more recently, on the bottom left, NIH has created a website to sort of centralize efforts in the replication and reproducibility space. And one of the efforts includes what's on the right, and that is the Replication to Enhance Research Impact Initiative. And in 2024, what they did is they gave out research supplements for third parties to replicate specific research experiments. And here we're talking about replication as in new data, new experiment. And on the bottom, they also recently came out with a challenge prize asking the community to come up with either ideas for important practices that should be replicated or experiments that would be high priority for NIH, and then also to identify people who had tried to do replications. And I'm pretty sure the winners of those should be announced sometime soon, but I don't think it's out yet. But of course we know there have been lots of large-scale replications, right? And this is just a smattering of them, but it spans the gamut all the way from cancer research all the way up to the SCORE project that just came out a couple weeks ago in the social sciences. And what have we learned from all of this? Well, not everything replicates. And a lot of times this is due to things like methods not being transparently reported. But it can also sometimes be due to confounding factors or chance or things like that. But what's interesting is that a lot of these studies, although the estimates vary wildly, there seems to be this central tendency about half of studies not replicating when you collect new data. And this has been labeled a crisis. But to evoke I'll say the ghost of statisticians past, and I hope Steve Goodman is okay with me making that joke because he is very much alive. This might not be all that far off from what we would expect. So Dr. Goodman found in this paper from 1992 that if you do an experiment and you get a result with a p-value less than 0.05, then if you repeat that experiment, and that's assuming that you measured the effect size correctly the first time, you actually only have about a 50% probability of reaching a p-value less than 0.05 the second time. That's just probability. And he wrote that in the medical literature, the failure of an experiment to repeat statistical significance achieved by previous studies causes concerns. This concern is based on a fundamental misunderstanding of the p-value. This confusion blurs the division between concepts of evidence and error for the statistician, and it obscures it completely for nearly everyone else. And this doesn't even get into how we're even defining a quote-unquote successful replication, and there are multiple ways to do that, or whether it even makes sense to repeat an experiment once and label that experiment as replicable or not, as like it's this binary. Because if we go back to Ronald Fisher, doing an experiment once only tells us so much. So I'm just saying, let's use with caution. When we're interpreting replicability, we just need to make sure that we're being careful with how we are doing that. Now, where am I going with all this? Ultimately, I'm just asking that the scientific community, whatever the field, just not forget about these earlier stages in the research process, because otherwise all the cool new tools that we're developing could be spitting out outputs that are just built on a house of cards, essentially. And this did not show up how it was supposed to show up either. But I wanted to use this slide to say that I am not in any way like laying blame anywhere. We exist in this ecosystem. I talked earlier with publication bias about the publish or perish culture. Scientists are under various pressures to publish in fancy journals, to get funding, and then for promotion and tenure committees, of course, taking all those other things into account in order to get that career advancement. So I see this more as a cultural issue than like an individual scientist issue. And this interconnected system can really make it easier or harder to do good science. And so I believe that all of the entities in the system really need to work together to try to incentivize good research practice. And this is just a slide about some of what NINDS is doing in the space. We're building an educational resource. We have various resources online, and we've had funding opportunities. Most notably, we've had a Rigor Champions Prize, so just identifying people who are trying to promote culture change in this space and rewarding them for that. And then also did not show up properly. Let's just focus on the thing on the left. So going back to what we as individuals can do, we can really just do what we can with what we have to try to improve research quality where we can. It doesn't matter where we are on this scale, we can do what we can to shift it all the way to the right if possible. All of us can find one thing today that we could do better tomorrow. And even if we're 100% perfect, which nobody is, we could be using that time to teach others, for example. And what matters is that we're sort of just on this journey to do the best that we can with the information that we have, and that small steps by a lot of people can lead to big change. And with that, I thank you for your attention. I hope my humble little biomedical perspective was in some way interesting to you, and I can take questions with whatever time we have left. Yeah. We have microphones in the back.
Thank you so much. That was a tour de force,
yes.
And I really appreciate it. Especially, a lot of it was eye-opening, but especially the relationship between the rigor practices and effect sizes, and infuriating to think about all of the wasted clinical trials. And so I appreciate that you guys are doing the things you said on the last slide about education and promoting best practices and incentives. But it seems to me the best way that NIH can change things is by saying you don't get your next NIH grant unless you do those things on your current grant. And I just wonder, right, and so like when I've never applied to an NIH, but I do NSF, and so we have to report the results of our previous NSF. And so I don't know if you do that, if you can do that, but also if there's capacity to do that, because I know, right, that researchers, they want to get their grants, so they'll make it look like they did. And so really to do the review, somebody would have to go through, maybe like Ted's AI thing can like do it for you, but somebody would have to go through to see if they really did it. So I don't know if you could talk a bit about using NIH's kind of, not its carrots, but its...
Well, I'll remind you, I'm at one institute at NIH. I don't have the power of all of NIH, and I can't speak for all of NIH. But there have been efforts to improve the peer review guidelines and what gets assessed in grant applications, and they updated them recently to have one entire factor, as they're calling it, entirely on rigor and feasibility. And so there has been some beefing up of the review practices. We have a program at NINDS where we take an investigator's prior publications into account in terms of how rigorously they've reported that they've done their experiments when we're deciding for like special awards that we give. And so that's something we at NINDS have tried to do. It's interesting you talk about the AI because there's been a lot of thinking in this space, and as of right now, it's not all that great at detecting everything we would want it to detect to say whether or not all of these practices are actually being performed or reported in some of these studies. There are a lot of sort of, inaccuracies that get into those, and it reports things that aren't actually there or misses things that are super important. And it really depends on the practice. Like some things like randomization are much easier to find, whereas things like proper exclusion of data sets is much harder to find. And so we're just not there yet with the tools to do that. Yeah.
Thanks so much. Really helpful and interesting to sort of hear how this works in a biomedical context. I was curious if you have just any initial thoughts sort of thinking about moving over into the social science world, how some of those earlier steps of like sort of building a good foundation work when you are working with like observational data or administrative data or types of data where you aren't controlling how they're generated or collected. Like just if you have initial thoughts of like what it looks like to be sort of transparent and rigorous in your treatment of those types of data.
Yeah, so if you have absolutely no control over the data collection, it does get really hard, but that's where sort of encouraging the original data collectors to be fully transparent about what did or did not occur during that data collection can really help the people on the other side. And that's why I really promoted that practice. But all you can do is say that we did or did not have information X, about the data set. And so that might be a caveat of your data set that you don't know whether or not a specific practice was followed properly when the data were originally collected. And so I lean towards transparency always being the best policy because there are going to be caveats. There's no such thing as a perfect study, right? It's just a matter of identifying where some of those biggest issues could be occurring and being fully transparent about that to do, like I said, to be able to interpret the data in its appropriate context. So you just do what you can. Yeah.
Hi, really enjoyed the talk. This question might be kind of out of deep left center field, and feel free to kind of answer it with one sentence if that's the case. But is there any, you brought up Fisher, is there any interest or talking about using more of a Bayesian framework for kind of this whole approach? And I'd love to hear your kind of, I don't know, is that an active area of debate? Is it have all the same problems that just standard kind of frequentist statistics has, or does it present other opportunities?
Yeah, I would say that yes, it is an active area of debate. There is a lot of work going on in that space. I wouldn't say it has all the same issues. It has different issues. There are particular decisions you can make with Bayesian processes that, can sometimes be a little arbitrary if you don't have the proper information that you need for those priors, right? And so it has its own issues, but yes, there's definitely active debate in how that could potentially help some inferences in science. Yeah. Mm-hmm.
You noted the powerful influence of publication bias in limiting what sorts of results we see, and the tendency for null results to be buried. The encouraging journals to publish more boring, uninformative, inconclusive results seems like a message that they might not be so enthusiastic about taking up. I wonder what you can say about building in systems, particularly through funding agencies like NIH, that require all funded research to be reported in some form or other, made available online, on preprint servers or other forums.
Yeah, so with the data management and sharing policy, they at least try to do that with the data, not necessarily with the publications. And there's always this argument, some pushback from the community being like, well, we can't get it published anywhere, so why? Why would you require us to do something that we actually can't do? And you're putting people in a double bind. Dr. Bhattacharya has mentioned about the possibility of investing more into platforms where you could share null results, but none of that has been done yet. And even if it does exist, we know from prior efforts to build platforms like this that it doesn't by itself fix the issue, because even when it exists, even when it's low cost, people still don't submit to it. And so that's where it goes back to this whole ecosystem issue, where you need to be incentivizing them to put the work into sharing it, or else even building the platform for them to share it, they won't share it. And so it's sort of this circular thing where everybody's pointing at somebody else to fix something first, and we all need to sort of fix a lot of issues at the same time. Yeah. Yeah, so a lot of that is considered sensitive data, so that's not really my area of expertise of whether we can do that. But we do, for example, promote preprints, as something. You can report that in your grant applications. Yeah. Is that it? Okay, all right. Thank you.